Hello and welcome at Basics of Biology. In this video series, we are discussing about enzymes for DNA manipulation. In our last video, we have already discussed about nucleases, and today we are going to cover another manipulative enzyme, that is the polymerases. Polymerases are the enzymes which are involved in the synthesis of new DNA strand complementary to an existing DNA or RNA template. Different types of DNA polymerases are used in gene manipulation. First is DNA polymerase 1, the Klenow fragment, reverse transcriptase, and Taq DNA polymerase. We will discuss these polymerases one by one. Coming to first DNA pol 1. DNA polymerase 1 is an enzyme that participates in the process of prokaryotic DNA replication. It was discovered by Arthur Kornberg in 1956. It was the first known DNA polymerase. It was initially characterized in E. coli and is ubiquitous in prokaryotes. DNA polymerase 1 has a dual activity, that is, it can act as a polymerase and nuclease. The 5' to 3' DNA polymerization activity, the polymerase adds complementary DNTPs on the template DNA. For this process, the polymerase requires a primer that can be extended in the 5' to 3' direction. This process occurs in the presence of magnesium ions. The hydroxyl group at the 3' end loses its hydrogen and forms oxyanion, and in the presence of magnesium ion, the oxyanion reacts with the alpha phosphoryl group of the DNTPs, then completes the reaction. DNA pol 1 has both the 3' to 5' and 5' to 3' exonuclease activities. The 5' to 3' exonuclease activity means that the polymerase is going to remove the nucleotides in 5' to 3' direction. This occurs when the polymerase identifies small single-stranded cuts, which are also called as nicks. The 5' to 3' exonuclease activity and 5' to 3' polymerase activity occurs simultaneously. The polymerase 1 replaces some of the nucleotides of DNA is called nick translation. Third type of activity of DNA pol 1 is 3' to 5' exonuclease activity. This activity is useful when the polymerase adds wrong nucleotides in the DNA. The 3' to 5' exonuclease activity halts the polymerase and removes the wrongly added nucleotide. This avoids mutation in the newly replicated DNA. The polymerase and nuclease activities of DNA pol 1 are controlled by different parts of the enzyme molecule. The nuclease activity is contained in the first 323 amino acids of the polypeptide. So removal of this segment leaves a modified enzyme that retains the polymerase function but is unable to degrade DNA. This modified enzyme is termed as Klenow fragment. Klenow fragment can synthesize a DNA or can seal a nick, but it cannot degrade the DNA strand and synthesize a new one. So its activities are, it can polymerize DNA in 5' to 3' direction. It has an exonuclease activity, 3', 5' exonuclease activity, but it does not have 5', 3' exonuclease activity. Reverse transcriptase, also known as RNA-directed DNA polymerase. It is an enzyme that is involved in replication of several kinds of viruses. Reverse transcriptase is unique in that it uses an RNA template. The ability of this enzyme to synthesize a DNA strand complementary to RNA strand is central to the technique called cDNA cloning. Another type of template-dependent DNA polymerase is Taq DNA polymerase. Taq polymerase is an DNA polymerase derived from a thermostable bacterium, Thermus aquaticus. It operates at 72 degrees centigrade and is reasonably stable above 90 degrees centigrade and is used in polymerase chain reaction. It has 5' to 3' polymerase activity and a 5' to 3' exonuclease activity, but it lacks the proofreading activity. This was all about template-dependent DNA polymerases. Thank you for watching this video. Please like and subscribe to this channel.